Hello guys, this is Jyoti Rana. Hope you all are doing well. Today I am going to teach you endomembrane system in eukaryotic cells. So let's begin. Many of the membrane organelles are considered together as an endomembrane system because their functions are coordinated. Endo means within. Within the cell membrane. Within this cell membrane, different organelles are works together. These are some of these are modified packaging and then transport that molecule in a coordinated way. It includes Golgi complex, lysosome, and vacuole. Functions of the mitochondria, chloroplast, peroxisome are not coordinated with any organelle, so these are not considered as the endomembrane system. Endoplasmic reticulum. Endoplasmic reticulum is the first part of the endomembrane system. It starts from the outer membrane of the nucleus, and it consists of network of disc and tubules like structure. This is the area of rough endoplasmic reticulum and this is the smooth endoplasmic reticulum. Ribosomes are present on the cytoplasmic surface of the rough endoplasmic reticulum. These are the ribosomes. And the area inside the disc or tubules is known as lumen. Intrinsic lumen, and this is external lumen, luminal area. External cytoplasmic area also called. And this is the smooth endoplasmic reticulum. Unlike rough endoplasmic reticulum, ribosomes are absent in the smooth endoplasmic reticulum. The presence of a network or reticulum of teeny tubular structures scattered in the cytoplasm, that is endoplasmic reticulum. Endoplasmic reticulum divided into the intracellular space into the two distinct compartments, luminal inside ER, extraluminal cytoplasmic compartment. Rough endoplasmic reticulum bearing ribosomes on their surface. So these are known as rough endoplasmic reticulum. These are extensive and continuous with the outer membrane of the nucleus. Functions: these are actively involved in the protein synthesis and secretion. We know that ribosomes makes proteins. These proteins are transported to the luminal side for the modification. Smooth endoplasmic. Absence of ribosomes are known as smooth endoplasmic reticulum. Smooth endoplasmic reticulum is the major site for the synthesis of lipid. In animal cells, lipid like steroidal hormones are synthesized in SCR. So रफ में क्या होगा, राइबोसोम्स प्रेजेंट होते हैं तो प्रोटीन सिंथेसिस होगा. स्मूथ में राइबोसोम्स एब्सेंट होते हैं, स्मूथ में लिपिड सिंथेसिस होता है. Detoxification of medication and poisons, storage of calcium ions, like muscle cells. Muscle cells में smooth endoplasmic reticulum ज़्यादा होता है as compared to rough endoplasmic reticulum because of muscle cells को calcium ions की ज़रूरत होती है. So like liver cells, unlike liver cells में क्या होता है, प्रोटीन और एंजाइम फॉर्मेशन होती है, तो वहाँ पे रफ एंडोप्लाज्मिक रेटिकुलम की रिक्वायरमेंट ज़्यादा होती है. तो लीवर सेल्स में रफ एंडोप्लाज्मिक रेटिकुलम ज़्यादा होता है. Then Golgi bodies. This is the structure of Golgi bodies. This is the cis phase, also known as convex or cis. The shape is the convex. This is also is receiving, also known as receiving side. Before the discuss about the structure of Golgi bodies, we should know how the endomembrane system works. So, firstly, come to the point of how the endomembrane system works. This is nucleus. This is RER, rough endoplasmic reticulum. Ribosomes are present on their cytoplasmic side. This is the lumen. The proteins are formed by the ribosomes are transported to the luminal side for the modifications. The modified protein come towards the Golgi bodies, and smooth, small patches are present in the rough endoplasmic reticulum. These small, why the जो राइबोसोम्स हैं वो यहाँ पे प्रेजेंट नहीं होते, छोटे छोटे स्मूथ पैचेस प्रेजेंट होंगे रफ एंडोप्लाज्मिक रेटिकुलम में. These smooth patches are formed vesicles. These firstly these are budding off, then form small sphere membrane vesicles. These vesicles these are vesicles. These vesicles transport this protein, modified protein, to the Golgi bodies. This is the structure of Golgi bodies. This is the cis phase, also known as receiving phase. The Golgi bodies receive vesicles, receives modified protein through the vesicles. These vesicles are fused with the cis phase, and then the proteins come into the cisternes. These tubular-like structures, sacs, these are the sacs, are known as cisternae. Already modified proteins, the sorting and tagging and packaging in the Golgi apparatus, and then transport to the trans phase. This is the trans phase, also known as maturing phase. Maturing phase. These modified proteins then again budding off with the vesicles and distributed to different organelles like lysosome or to the plasma membrane. This is the all about endomembrane system. Come to the structure of Golgi bodies. This is the incoming transport vesicle fused with the cis phase of Golgi apparatus and transport the protein to the lumen of the cistern. Then, after the modifications, these proteins are transported through the vesicles. These are newly formed vesicles and distributed to the different organelles. This is all about endomembrane system and Golgi bodies. Consists of many flat disc-shaped sacs, cisternae of 0.5 micrometer to 1 micrometer diameter. These are the cisternae, diameter of 0.5 to 1 micrometer. And this is these are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis. This is the area, convex cis, or forming phase and concave trans and maturing phase. This is concave trans and maturing phase. Forming phase is also known as receiving side of the Golgi apparatus. The cis and trans phases of organelle are entirely different but interconnected. These are different in shape and their functions are different, but these are interconnected. Functions of the Golgi bodies: perform the packaging material to be delivered either to the intracellular targets or secreted outside the cell. Means different organelles में भी जा सकती हैं और secrete out भी हो सकती हैं. Material to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the endoplasmic reticulum. So ये इतने ER के इतने close क्यों होते हैं? क्योंकि जो material होता है जो transport हो रहा है वो through vesicles in the form of vesicles ER से come to the cis phase of Golgi apparatus, fuse with them and move towards the maturing phase. Then they are distributed to the different intracellular targets or secrete out. Then a number of proteins synthesized by the ribosome on the endoplasmic reticulum are modified in the cisternae of Golgi apparatus. In the cisternae of Golgi apparatus, in the lumen of the cisternae, the proteins are modified if they are sorting, tagging and packaging what they are required before they released from the transphase. Golgi apparatus is important site for the formation of glycoproteins and glycolipids. Some proteins are tagged with the carbohydrates, some lipids are tagged with the carbohydrates. So protein with carbohydrates known as glycoprotein, lipids with carbohydrate known as glycolipids. These modifications are occur in the Golgi apparatus. Then come to the lysosome. Lysosomes are the membrane bound vesicular structure. Vesicles की तरह होते हैं, membrane bound structure formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all the hydrolytic enzymes. Hydrolytic enzymes present होते हैं. pH acidic होता है. pH acidic. Hydrolytic enzymes are hydrolases. Lases मतलब जो ब्रेक करते हैं, breakdown of protein, lipid and carbohydrates. So if they are lipases, they break lipids, proteases break proteins and carbohydrates break carbohydrates. Optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. And that's all. Vacuoles. Vacuoles are also lysosome-like structure. Lysosome-like organelle. These are present in the plant cells. The vacuoles in the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product and other material not useful for the cell. Vacuoles में जो material present होता है वो cell के लिए useful नहीं होता. The vacuoles is bound by a single membrane called tonoplast. This is the vacuole and the membrane, single membrane is known as tonoplast. Tonoplast facilitates transport a number of ions. Facilitates transport number of ions. So ions move towards inside the cell, inside the vacuole, sorry, not cell. In plant cell, vacuoles can occupy 90% of the volume of the cell. In plants, tonoplast facilitates the transport of a number of ions and other material against concentration gradient in the vacuole. Against the concentration gradient. It means from low concentration to high concentration. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. This is the cell. This is the vacuole, cytoplasm. So, the concentration gradient of ions is higher in the vacuole and low in the cytoplasm because the tonoplast facilitates the transport of ions against the concentration gradient. In amoeba, contractile vacuole is important for osmoregulation, excretion in many cells as in protist food vacuoles. are formed by engulfing the food particles. So ये as a food vacuoles osmoregulation और excretion की तरह भी काम करता है vacuole. So this is all about endomembrane system. In the next video, we discuss about the organelles like mitochondria and chloroplast. So stay tuned and hope you guys liked my video. So do like, subscribe and share my channel. Thank you so much.